Hello guys, I am Shubham from Fluid Tutorials. This video is about photoreactivation. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. In the previous video, we discussed about mismatch repair pathway. This video is about photoreactivation. Basically, what is photoreactivation? Why photoreactivation is used? So it is used in the case of pyrimidine dimer formation. We have discussed this as a DNA damage, right? Thymine dimer repair. Okay, fusion of pyrimidine dimer, right? And this actually is an example of direct reversal of DNA damage. So let's see what is first of all dimerization. See, this is a thymine dimer. Okay, basically pyrimidine dimer formation takes place. Okay, this is thymine dimer, right? Two thymines adjacent to each other get paired to each other. We can see the cyclobutane ring formation takes place here. This is a ring formation takes place. Alright, and due to this formation, they are paired to each other. So thus they cannot pair with the what we can say complementary pairing. They do not show complementary pairing because they are paired to each other. Right. So this is a pyrimidine dimer. Pyrimidine dimer. So it can happen between right TT, CC, and also between TNC. Okay, this cyclobutane ring formation. Right. So now how this is repaired? This is repaired by the process of photoreactivation. So here comes the enzyme DNA photolase. DNA photolase. We can see here pyrimidine formation is due to UV light, UV radiation. Right. So this is a pyrimidine dimer. Okay. Now this has to be repaired, done by DNA photolase. Okay. DNA photolase. Photo means light. So it also requires light. So see, it gets bind to this timer. We can see here with the help of visible light. It captures energy from the light, and then the energy is used for breaking the bond between these dimers. Okay, see, we can see here light has been used to break the bond, covalent bond between the dimers, and after the bond has been broken, the lyse is released out, and we can see this is repaired. Right. So what we can write here, DNA photolase captures energy from visible light to break the covalent bond. Covalent bond means what? Cyclobutane ring. In the pyrimidine. Dimer. Here the case is thymine dimer, right? Now there is one more example, damage base, that is methylguanine, ortho 6 methylguanine. So this is a product of methylation taking place in the guanine, right? So what happens now? This has to be repaired. So here comes the enzyme methyltransferase. What will methyltransferase do? This will remove this methyl group from guanine and it will give it to the cysteine residue. So here will be cysteine, right? So methyl is transferred from guanine to cysteine. See, we can see here. Enzyme gets bound to guanine and then will be released out taking the methyl group. And hence this is repaired. Right? These two are the processes of direct reversal of the DNA damage. So what can we write here? A repair done by methyltransferase. Transfer of methyl group from guanine to cysteine. That's it. And thus the methyl is removed and guanine is normal now. Okay. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.